I want to start out by saying that the work I'm going to talk to you about is not my work. It's work that's been done by a large collaborative group. Some of the people who I'm working directly with in Seattle, in Leiden, in Rochester, and at companies are listed here. But it's also work based on a compilation of studies done by other people who will be talking to you today and by other researchers in the FSHD community in an effort not to just talk about what I've done, but really to get across a general sense of where we are in FSHD. Now, I usually talk to scientific audiences, and so I spent a lot of time really trying to think about what I would like to convey to this group today, and there are two things. One is I can say just very straight out, I am very excited by the amount of progress that's occurred in the last 2 years in FSH research. I spoke to this group 2 years ago, and I think at that time we were just beginning to understand the mechanisms of the disease, and that's been solidified over these last 2 years. And I think, as I'll try to emphasize at the end of the talk, we're really at a point now where we know what to do to go forward and start developing therapies. And so I think that that's a real sea change over the last 10 years in FSH research, and I'm very happy to be able to be part of it. So in terms of what to convey to you today, I wanted to give you an idea of what causes FSH and why that gives us an idea of how to develop therapies for it. And so one of the things I talked about 2 years ago was this sort of attic or living room analogy. And so each cell in your body has a copy of all of your DNA. And that DNA is present in every cell, even though the genes encoded in each cell are only used by some cells. So neurons use some genes, muscle cell uses some genes, other cells, but nonetheless all of the DNA is there. And it's a lot of DNA. It's about a yard long or a meter long of DNA in each cell. And so the cell has to package up this DNA into a very small nucleus. It's about 0.1 millimeter in diameter. And so it has to then find the genes it wants to turn on and not find the genes it wants to keep off. And so the genes that it doesn't use in any given cell are just packed up really tightly. It's sort of like when you take your stuff and you stick it in the attic in a box that you don't want to use, and it's packed as tightly as you can in the box, and genes that you want to use are unwound and open and sort of like in the living room on your coffee table or something like that. And that's shown pictorially here, where this is an electron micrograph of a cell. The outline of the boundaries of the cell are there, and then there's the cytoplasm. And here's the nucleus where all the DNA is wound up. And you can see there are dark areas and light areas in the nucleus. And the light areas are where genes are that are active and that are being used by the cell, and the dark areas are where the cell puts genes that are off. Sort of like that's the attic, right, and the light areas are the living room, and the terms we use for them are the dark areas are heterochromatin and the light areas are euchromatin. That's just the term for the dark off areas versus the light on areas. And that's pictorially shown here, where the sort of purple line represents DNA being wound around the proteins that package it, and on the left are areas where genes are expressed. They're loosely packaged, and on the right, their genes are repressed. They're in the attic, and they're really tightly packaged up and less accessible. So again, when a stem cell is growing, a stem cell that will give rise to lots of other cells, it has lots of genes on, and lots of genes are in the living room. And then when it differentiates, so for example, if it differentiates to a cardiac cell, a lot of the stem cell genes get turned off. They're put in the attic, or they're put in heterochromatin. And usually you don't go back. You don't go back from a differentiated cell to a stem cell. And part of the reason is there's a nice lock. So you put it in the attic, and then you lock the attic door with a modification of the DNA called methylation. And it's been shown that if you don't have this lock, you can slip out of the attic. and cells can turn on genes that aren't supposed to be on. And so again, during going from a stem cell to a differentiated cell, you take lots of your stem cell genes, you put them in the attic, and you lock that attic door. So what happens when the attic door isn't efficiently locked? This slide shows a series of mice, all of which are genetically identical. But the lock that turns on the coat color in this mouse is not very efficient. It has a rearrangement in the genome in all of these mice that make the lock less efficient. And so sometimes the gene is on, and sometimes the gene just doesn't stay on. It slips into the off position, and sometimes it's intermediate, and you get this salt and peppered expression. So you get a variegated expression in the mouse when these locks aren't efficient and things can slip in and slip out of the attic. And so the variegated term is something you're probably familiar with plants and variegated leaves. And again, that's sort of slipping in and slipping out of that on-off position. In some cases, it's due to the fact that there's not a very good lock. So how does that relate to FSHD? You all are familiar with FSHD. It's an autosomal dominant disease, slowly progressive, onset about 1 in 20,000, predominantly early on affects the upper extremities, but often will go on to affect most of the genes, most of the muscles in the body. The mutation was mapped to the short end of chromosome 4, very close to the end, where there's a number of repetitive sequences, about 3.3 thousand nucleotides repeated head to tail many times. And in each of those repetitive units, the D4Z4 units, there's a gene called DUX4. DUX4 is a gene that's normally used in humans. It's normally expressed in stem cells that make sperm and oocytes, and this is a picture of the human testis, a section through that, and the brown color here indicates DUX4 expression, and the DUX4 is expressed in the stem cells of the human germline that will give rise to sperm or eggs. And then it's not expressed in healthy skeletal muscle tissue and somatic tissues, and it's turned off by being put in the attic and being locked there by DNA methylation, so it doesn't get re-expressed in skeletal muscle. So what causes FSHD? So now we know that, again, you have these D4Z4 repeats on chromosome 4 that have the DUX4 gene, and normally they're turned into heterochromatin in skeletal muscle tissue. But in FSHD, you have a deletion of those repeats, and so you have fewer repeats, and the ability to put it in the attic and lock the door is less efficient. You have less heterochromatin, less DNA methylation. And we know that it's this loss of heterochromatin and loss of methylation, this loss of being able to lock the door, that causes FSHD, because there's a less common form of FSHD, FSHD2, where you simply have a loss of the heterochromatin and a loss of the methylation without the contraction. And so the contraction makes the lock inefficient in FSHD1, and in FSHD2 there's another mechanism that makes the lock inefficient, and it's that inefficient lock that leads to the development of FSHD. And what it does is allow that DUX4 gene to leak a little bit in skeletal muscle. The attic door is not locked enough. And you get occasional nuclei, such as these pink nuclei here, where the pink is the stain for the DUX4 gene, and this muscle cell shown in green, where most of the nuclei in this are blue, they're not expressing DUX4, and an occasional nucleus will lose the ability to keep that attic door shut, and DUX4 will leak out and be expressed. And so what does it do? So we have a developmental model now of FSHD, where the DUX4 is normally expressed in stem cells of the germline. We think it has a normal role in those germline cells in activating stem cell genes. That when you go to differentiate to skeletal muscle, the heterochromatin silences that gene and locks it off. And then the mutation that causes FSHD decreases the efficiency of that lock. And so you get some misexpression or some leaky expression, variegated expression of this DUX4 gene in skeletal muscle. And we can show that that reactivates stem cell genes in skeletal muscle. It essentially tells the skeletal muscle cell I'm a germline cell. I'm not a skeletal muscle cell. I'm supposed to do something else. And this shows, this is the only data slide I'll show it. On the left, the control is muscle biopsies from unaffected individuals and then muscle biopsies from FSHD individuals, looking at genes that are normally only expressed in the germline that are activated by DUX4, and these genes are now misexpressed in skeletal muscle. DUX4 is misexpressed and activates the germline program in skeletal muscle. So why would that cause a disease? And I think from there we have a number of viable mechanisms for how this damages skeletal muscle. One is activating a germline program in another tissue could in itself be bad. It could just be incompatible with the program that's normally expressed in skeletal muscle. And many genes are expressed that could cause muscle cell death or muscle wasting that are normally expressed in the germline, but when misexpressed in skeletal muscle can cause a problem. There's another potential mechanism that's very intriguing to us because the testis and the germline is immune privileged, just like the brain. And so proteins that are expressed only in the germline, when they're expressed in skeletal muscle or other cells, can induce an immune response to those genes. And so it's quite possible that the re-expression of these genes is inducing an immune response against the skeletal muscle. And that's intriguing because FSHD pathology has always been associated with an inflammatory component with the immune cells, CD4 and CD8 T cells, part of the immune system, infiltrating into the skeletal muscle. We don't know this, but our hypothesis at this point is that they might be attacking the muscle cells because of the re-expression of some of these germline proteins in the skeletal muscle. So I just talked about two possible mechanisms from the activation of the germline program that could cause FSHD. Another one that very much intrigues us is that one of the genes that, again, DUX4 normally expresses in the germline and misexpresses in the skeletal muscle is a defensin protein. It's a small secreted peptide, and that peptide binds to a receptor that's necessary for muscle regeneration. This panel in the left shows phase, and on the right, a staining for a muscle protein of normal muscle cells undergoing differentiation where they fuse to form multinucleated myotubes and express their muscle markers. But if you add this defensin peptide that's induced by DUX4 to those cultures, these cells no longer fuse, they no longer differentiate. And so the expression of DUX4 not only can damage the muscle itself, but it also induces the secretion of a small protein that blocks regeneration of the muscle. And then finally, something that we are interested in but we don't really understand is DUX4 activates the expression of endogenous retrotransposons. These are ancient viruses that riddled our genome at the root of the primate lineage, and those are re-expressed, and we're very interested to know whether that re-expression contributes to the disease. So I've just gone through a number of mechanisms and the model for how the re-expression or the leak expression of DUX4 causes FSHD. The good news is this gives us a lot of understanding about how to develop drugs to treat FSHD. And one sort of top tier is to shut off the DUX4 leakiness. This is a gene that is almost entirely off, and we just have to help it be off a little bit more. And so we can try to develop mechanisms that enhance that heterochromatin repression, sort of make the lock on that attic door a little bit more efficient. And there are standard ways of doing that. Either shRNAs or siRNAs can also target the mRNA and block its expression. We can try to develop drugs to block DUX4 protein activity so it doesn't activate the genes that cause the problem downstream of it. Or we can interfere with each of these mechanisms that I talked about. Try to interfere with some of the germline programs, try to interfere with the immune response, try to improve the regeneration. So we have a lot of work to do, but we know what to do. We have a good roadmap for how to go forward now. And then finally, we have good biomarkers for following this. We have targets of DUX4 that we can look at and know if these therapies are having any benefit on what we want them to do. I just want to say one quick word about how we know that our level of confidence, something that increases our level of confidence that the expression of DUX4 is causing FSHD, and that has to do with FSHD permissive and non-permissive alleles. One of the things I want to point out is FSHD is a retrogene, which means that it was a cDNA, or an mRNA that was retrotransposed and moved to a new location in the genome. Without going through all of the details, when that happens, the new gene, the retrogene, is divorced from the evolved regulatory elements that regulate its expression and when and where it's expressed. And also this site outside of the gene that's called the poly-A site, which is a marker for stabilizing the RNA. And so usually retrogenes mutate away and become junk in the genome unless they can pick up new polyadenylation sites or new enhancers and promoters. And the DUX4 gene did that on chromosome. There are copies of DUX4 on chromosome 10 and chromosome 4, and they picked up this polyadenylation site that's outside of the D4Z4 repeats. And in the germline, there are two different regions that can be used for polyadenylation. I'm having a hard time with my arrow here. One of them in the top is used by either chromosome 10 or chromosome 4. It's really distant to the DUX4 retrogene, and that's used in the germline. But in somatic cells, it's not available. In skeletal muscle, part of the lock is that polyadenylation site is just not available. There are, however, variants of chromosome 4, which I'll just call chromosome 4QA here, that have a slightly different sequence in this region called 3 that creates a new polyadenylation site. And so that can be used in somatic tissues. And so when you have FSHD, if you have a contraction on this A allele that has this critical region that has the polyadenylation site that the others don't have, you get FSHD. Whereas if you have contractions on the other alleles, the non-A allele on chromosome 4 at chromosome 10, you don't get the disease, indicating that this polyadenylation site and the stabilization of the DUX4 RNA is necessary. And so what that polyadenylation site does on the left, if you have it, you stabilize the RNA, and if you make the protein on the right, you don't stabilize the RNA, you don't make the protein. And this finding really gives us a lot of confidence that it is the leakiness of the DUX4 that is responsible for some of the major components of FSHD. And I just want to close with one thing that I think is very speculative, and I want you to realize that this is something that I'm thinking about as opposed to something that we know is true. But the DUX4 gene, again, retrotransposed and moved itself to a different region of the genome. And one of the things that's interesting is it didn't become a pseudogene. It didn't mutate away. We've kept the retrogene. In fact, we've lost the parental gene that DUX4 arose from. And so that suggests that DUX4 is doing something for us. And DUX4 retrotransposed at the root of the primate lineage, and so it presumably is doing something for primates. There's a lot of precedent for retrotransposed genes driving a new phenotype. And so this is a slight digression, but dogs that have short legs, like corgis and basset hounds and dachshunds, all have short legs because of the retrotransposition of a different gene, a growth factor receptor that retrotransposed into a region, so now it was misexpressed in the developing legs, and it stunted the growth of the legs. And so all of these dogs were bred from this same single event. And so retrotransposition can drive evolution. It can drive the evolution of a new phenotype. And I think it's interesting to note that DUX4 retrotransposed at the root of the primate lineage. And so all primates have this retrotransposed DUX4 gene. And what does the phenotype of FSHD suggest? It's a weaker facial muscles and weaker upper extremities. And one of the things we're thinking about is maybe this is a phenotype that was actually selected for. And this is what DUX4 is doing for primates. It's giving us a weaker facial muscles, more expressive ability to talk, to smile, compared to non-primates, weaker upper extremities because we walk on our hind legs. And the price we pay for having this advantage in the primate lineage is if that phenotype is a little bit worse, if it just goes a little bit too far, we get something that we call a disease, FSHD, because it's somewhat more of an extreme phenotype than what we're actually selecting for in the population. So again, something to think about in the notion that FSHD might just be a little bit more extreme example of what primates really selected for during evolution. Okay, I want to acknowledge some of the agencies, including the FSH Society, that were critical in supporting our work and other work in this area. Thank you.